DNA is replicated? Well, this is how that replication occurs. And DNA replication is going to depend on specific base pairing. And by that, I mean that A is always going to go with T and C is always going to go with G. So DNA replication is going to begin with separation of the two strands. And then enzymes use each strand as a template to assemble a new strand. So DNA follows a semi-conservative model because the new strands, one will be old and one will be new. Okay, so let's look and see what happens. I'm going to change my pen to the magical one where it disappears. First, we start with parental DNA. This is our molecule. And the first thing that's going to happen is it's going to separate. And now we've got two templates. We've got a template on this side, and we've got a template on this side. After they separate, we can add these nucleotide bases in here, and we can make a new strand of DNA. So now we have an identical strand. These are two identical daughter molecules that are formed, and you'll note that they have the same pattern. We started with ACG. We end up with ACG, that's the conserved strand, but the new strand also is ACG, and its partner, TGC, remains TGC. So we're able to reproduce our DNA. DNA replication proceeds in two directions at many places. Oops, I need to change my. Two directions at many sites simultaneously. Many. Okay. So we're going to look at this in just a few minutes, but not right now. See if we can get this to work for us. We've got an enzyme DNA polymerase. That's able to synthesize the daughter strand in one piece. And the other strand is synthesized as a series of short pieces that are then put together. Now, the reason for this somewhat complicated problem is that the DNA polymerase can only add to the 3' end. And if we look at our DNA molecule, it has a 5' end. It goes 5 to 3 this way, but the complementary strand goes 5 to 3 this way. So if you can only add onto the 3 end, you can only add to this end, and you can only add to this end over here. So the molecule grows in the 5 to the 3' direction. You can only add to the 3'. So the way the molecule what's the solution to this problem is that the DNA is split. This is a DNA polymerase, and this strand is made continuously. It's 5 to 3, and it could go and work continuously. On the other strand, shorter 5 to 3 sections are synthesized and then put together with the ligase. The ligase down here connects the short strands that are not contiguous. Okay? Multiple origins of replication serve to shorten the time necessary for DNA replication. Chromosomes are very, very long, and by having multiple origins of replication, we get, it speeds up the process. Okay, now I've got this video to watch. So during replication, DNA acts as a template for both the formation of new molecules. So you could see the DNA coming in here, and this is the new DNA coming out. New DNA strands are synthesized by a multi-enzyme complex. Here, one strand is leaving from the top of the frame. The other new strand is leaving from the bottom of the frame. The first step in DNA replication is the separation of the two frames. The enzyme called helicase. It spins the DNA to unravel it, 10,000 RPMs in the case of bacterial systems. The separate strands are called the 3' and the 5', and distinguished by the direction in which their component nucleotides join up. The 3' DNA strand, also known as the leading strand, is diverted to DNA polymerase and is used as a continuous template for synthesis. That's the one that I said does continuously. The other half of the DNA, as known as the lagging strand, has the opposite orientation and consequently requires a more complicated copying mechanism. It emerges from the helicase, the lagging strand, is organized into sections. These are then presented to a second polymerase, the enzyme in the preferred 5'3" direction. These strands are essentially done backwards, and copying is complete, it's released. So that it does it in big loops so that it can go in the 3' to 5' direction the way it needs to do that.